Well, first, I want to thank everyone for joining us tonight. As you know, The Cure Starts Now is dedicated to funding pediatric brain cancer research to find the home run cure for all cancers, but we can't do it alone. And we absolutely believe in partnering with others that have the same determination. And that's what brings us here tonight. So we want to highlight a new partner of ours that's helping to advance research. Before I make the formal introductions, I do want to mention we're going to be using our chat feature tonight. So if you have questions, just wait to chat to the end so it doesn't distract them. But I will be asking the questions at the end. So to get started, it is my pleasure to introduce you to Chris Rivera. He is the President, CEO, and Chairman of Emulate Therapeutics. And we have Dr. Javier Figueroa, who is the Vice President, Preclinical Development of Emulate Therapeutics. They're here to talk a little bit about their experimental HALO device that's used for the treatment of DIPG, DMG, and really how it works and the potential to help patients with brain tumors. So why don't I go ahead and hand it on over to Chris Rivera.
Thank you very much, Melissa, and good evening, everybody. Thank you for taking some time to join us and learn a little bit more about Emulate Therapeutics, our technology, and how we hope to be able to help patients and children with not only pediatric brain cancer, but adult brain cancer as well. As Melissa mentioned, I'm Chris Rivera, I'm the CEO, and with Dr. Figueroa, who's our head of Vice President of Preclinical Research. So we will start the program now. And so we are a company based out of just outside of Seattle, Washington, about 20 miles south as we sit here in our lab in Kent, Washington. So we are a digital therapeutic company. We are using ones and zeros to try to treat maladies of the brain. We're beginning with brain cancer, both for adults as well as for children. We're using radio frequency energy, which is, I'll explain a little bit more about that momentarily, but radio frequency energy, or RFE, has been used for decades in healthcare. We're using RFE to create magnetic fields that emulate the mechanism of action of non-covalent drugs. And again, we'll explain that in a moment. We've been around over 20 years. We've been 100% funded by angel investors. And again, initially we're focusing on maladies of the brain, both brain cancer as well as mental health conditions. There's many advantages to our technology. We can, for the most part, emulate most drugs' mechanism of action. We can do it relatively cost effectively and relatively quickly to at least generate proof of concept data to see if it's worthwhile to go into human clinical trials. We have two global partners, one in Japan, Teijin Pharma, has licensed the rights to glioblastoma, and in India, Therapeutics has licensed the rights to both diffuse midline gliomas as well as glioblastomas. About 30 patents that have been issued so far, and we continue to add to our patent portfolio. As I mentioned, we're using radio frequency energy in healthcare. For the most part, RFE has been used on the high end, high energy, high ionizing end of the spectrum, gamma rays, X-rays. I think we're all probably familiar with that. We're working at the very opposite end, at the ultra low end, below 22,000 hertz. How we're able to create these wave files and magnetic fields is a device here that sits in our lab about 50 feet from where Javier and I are. It's called our acquisition system, or mid system. It's about a 4-foot by 3-foot lead box. We're using technology called SQUID technology, superconducting quantum interfering device. SQUID technology was created by our government during the Cold War for the most part. These are ultra-sensitive listening devices called magnetometers. So in our case, with our magnetometers and our SQUID technology, instead of using them to listen to conversations maybe in Moscow or Beijing, we actually use these magnetometers to listen to molecules as they are in this chamber. When we want to record a wave file or a wave file that would create a magnetic field to emulate a drug, for example, a chemotherapy agent, we supercool this device to about 4.4 degrees Kelvin using liquid helium. The inside of this chamber is quiet enough and it's cold enough that it's actually like being in outer space. Javier then and his group will take a chemistry. In the case of brain cancer right now, we're working with a chemotherapy agent called paclitaxel. He took samples of paclitaxel in various dilutions and put it in solution. And then in a chamber about the size of a lipstick tube, we drop that paclitaxel chemotherapy in solution. Mm-hmm. And over a period of seconds to minutes, the molecules in that solution begin to tumble and turn. And when they tumble and turn, they begin to leave a minute magnetic wake. So the analogy that I use is think of a canoe going down a canal. That wake that canoe is leaving is what this molecule is leaving is a magnetic field or digital signature of that molecule. The environment inside the the acquisition system is quiet enough, the magnetometer is sensitive enough, we can then measure and record that magnetic wake as a time domain series, meaning we now have millions and millions of data points, about 2.5 million data points just from that one recording of that single molecule. We then convert that into a WAV file. So again, the analogy I use is recording the song of a molecule, and then we can replay, and I will explain how we replay that and create a magnetic field that hopefully acts like the drug will act without the drug actually being there. And in this right box, if you look at how non-covalent drugs work, about 2/3 of drugs that have ever been approved by the FDA work non-covalently, meaning they don't actually bind or block to a receptor site. They exchange information, and we believe we're emulating that exchange of information with our technology in this case, a tumor site, a tumor cell, without the drug actually being there. And the tumor cell doesn't know if it's a drug there or if it's our magnetic field emulating the effects of that tumor cell, of that chemotherapy. So as I mentioned, we then, in this case, we took the recording, the song of paclitaxel, which is a chemotherapy agent that's been used for decades in cancer treatment very successfully. So here was here's the chemical structure of paclitaxel. Here is the WAV file or the song of paclitaxel. And then we download that WAV file to this controller, and I have one in my hand here. So think of this as your iPod or your cell phone, and when we turn the controller on and play that song of paclitaxel, in the case, this is a device that the patient with glioblastoma wear. Patients with diffuse midline glioma wear a similar coil, but on the back of their head. We create a magnetic field that's roughly the circumference of the coil, and it goes up and down about 15 centimeters in each direction. In the case of diffuse midline glioma, it is projected out basically to the front of the cranium. And we're creating a magnetic field of paclitaxel. And what we're seeing are tumor sites or tumor cells are actually interacting with that magnetic field of paclitaxel without the drug actually being there. And they're reacting as if the drug were there. And we have not only data in cancer agents, but we also have it now in psychedelic agents, pain models, et cetera, et cetera. So I'm going to pause there and hand it over to Dr. Figueroa to share with you some of the data that supports the hypothesis that we are creating that we can actually help children and adults with brain cancer. Javier.
Thank you, Chris. So what we're seeing right here is called a tubulin polymerization assay. So tubulin is made up of two proteins, and they're the foundational block for making microtubules. And microtubules are very important for cell division. Microtubules are critical for a cell to divide, and they're also critical for rapid division of cancer cells. So if you can actually block tubulin formation or actually cause a freezing of the tubulin, you actually have a very strong anti-cancer agent. So as I mentioned before, the tubulin polymerization assay gives us an idea see if we can actually emulate the mechanism of action of the drug itself, which is paclitaxel. So the green line, so the bottom of the line is basically your time. And then the up and down line is the growth of the tubulin into microtubules. So as time goes on, you can see that the line increases. The black line represents the normal background rate of tubulin polymerizing into microtubules. That's just basically the baseline. That's the normal rate. The green line is when you add the drug paclitaxel, and as you can see, it causes an increase or speeding up of the rate of tubulin formation, which is associated with causing cancer cells or rapidly dividing cells to freeze and not divide. That is, they're locked into place, and then they undergo apoptosis or necrosis. The two middle lines, the yellow and the red, those are our WAV files, our two signals of A1A. So the yellow line is our A1A that we've tested in the clinic, and the red line is an improved version of the A1A signal that we're currently testing in preclinical trials. So as you can see right there is that it's right in between. Both lines are in between the 50 nanomolar paclitaxel, which is roughly what tumors see when people are given chemotherapeutic agents. So we're right in between, roughly about 50% of the actual therapeutic dosage that is normally seen by tumors. So this shows that we can emulate the effects of paclitaxel itself just by using the magnetic fields. And this is great potential because when people are given paclitaxel, usually in 3-week blocks of treatment because of the toxicity, we're actually delivering paclitaxel constantly, 24 hours a day, 7 days a week. So it's actually a great advantage because we also don't have any side effects associated with the actual physical drug. The magnetic field delivers the effects of paclitaxel constantly, continuously, but it doesn't have all the usual side effects or severe adverse events that are reported with paclitaxel. And again, that's the best part of it. Now, one of the things that we are a DIPG company, we are committed to getting our technology and our treatment to treat the scourge of neuro-oncology. So we partnered and worked with UCSF to test the effects of our A1A signals and our A1A signal in a mouse model of DIPG. So normally one of the first lines of animal models is to test the effects of any chemotherapeutic agent or any therapy on the overall survival of the mice when they're implanted with these tumors. And as you can see, the red line represents our A1A signal, and the black line represents the control animals. As you can see, we do see an overall increase of 2 days of increased median survival and at least 3 to 4 days of overall survival, which is statistically significant. This corroborates what we've seen so far with our compassionate use program, which we had run in 2018, which we'll talk about right now. So again, we've been in talks with the Food and Drug Administration, and we've already had several meetings with them, looking at the potential to provide this technology as a humanitarian device exemption pathway to the FDA. FDA asked us to run a compassionate use program as a preliminary test of safety in this population. And again, we are now moving forward. We want to proceed with a pivotal phase 3 trial in this population. Overall, we've seen that with our GBM as well as our DMD compassionate use program, we have seen not only 3 months improved survival, but we've seen evidence that this can provide an increase time to survival. Now, if we look over to the right side of the screen, you see an image of an MRI, and you see all the lines indicating the strength of the magnetic field. The reason that we designed this the halo to be used at the back of the head is that way we can direct the majority of the magnetic field to where it's needed the most, which is the brainstem and the pons at the back of the head. So this provides the greatest coverage at the highest energy density to the area that it's needed most. And again, it's easy to use, it's safe to use, as we've tested not only in adults but in our early pediatric cases. And we think that this is going to be a great advantage. So the results from our compassionate use program, we treated a total of 16 children, 14 with DIPG and 2 with other brain cancers. We saw that that 12 out of our 14 pediatric cases that came in survived greater than 12 months using the Halo system. Again, we saw no device-related serious adverse events. We did see some adverse events. One patient reported some nausea, fatigue, and sleepiness, and there was some reports of vomiting in another patient. But overall, this was very encouraging in that there seemed to be a signal for efficacy, but there was definitely safety, not only in our pediatric patients, but also in our phase 2 GBM trial, which showed excellent safety profiles. Now, as I mentioned before, we have done a phase 2 clinical trial in glioblastoma, which is again another neuro-oncological tumor. As you can see on the right side of the graph, which is median overall survival, traditional therapies for GBM in adults shows that there's a 7.2 month overall survival. Our Voyager system, that is no chemotherapy, just the Voyager system alone, was equivalent to active chemotherapy. But what's interesting or what's encouraging is that when you add our Voyager system with best standard of care, which is chemotherapy and radiation, we see a 3+ overall survival advantage when we do this combination therapy. So this is very encouraging information, and it corroborates what we've been seeing not only in our animal models, but in our compassionate use protocols and all the preclinical data that we have so far. Now, what's encouraging, or what we've been working on, is that we're a platform technology. That is, we're not just using our technology to emulate paclitaxel. We can emulate a wide variety of different chemistries, different therapies. So, for example, if we wanted to go after epidermal growth factor, and we have preclinical evidence showing that we can actually affect epidermal growth factor receptor expression, we can either emulate the effects of something called small inhibitor RNAs, or we can emulate the effects of actual drugs. Farnesyltransferase is another target in DIPG. PDGFRA, platelet-derived growth factor receptor, is another target in DIPG. Vascular endothelial growth factor receptor 2 is another target in DIPG. Cyclin-dependent kinases, bromodomain 4, and for example, compounds like ONC201, which go after the dopamine receptors, those we can also emulate. We also have preliminary evidence in immuno-oncology. For example, CTLA-4 and PD-1 are major targets that have shown great promise in treating other tumors. We also have preclinical evidence showing that we can target those receptors as well. So because we have this versatility in being able to target not only genetic and metabolic pathways, the preclinical evidence in immuno-oncology in animal models shows that we can directly inhibit and also decrease tumor volume in animal models. So what we have right there is on the left side is basically our control group. White noise and untreated animals show the same growth rate. They're statistically identical. But when we expose the in this tumor animal models, the signal that will decrease CTLA-4 and PD-1 expression in tumors, we see a decrease in tumor volumes, as well as increased survival in our animal models. So we have many different pathways, many different targets that we can hit, and we feel very confident that as we progress and learn how to treat DIPG effectively, we can hit multiple targets at the same time. And as I mentioned earlier, we do have preclinical evidence showing that we can actually directly target epidermal growth factor receptor. So on the left side, we have the survival curves of an animal model in which we specifically targeted EGFR. That aqua line is mice that are exposed to the small inhibitor RNA against EGFR. That is, we're actually targeting to reduce the overall expression of EGFR. And then the black and gray lines are an untreated group and a signal that played just a nonsense or scrambled siRNA target. On the right side, we have the staining. The furthest left image at the top is EGFR labeling in our untreated. Middle one is a scrambled signal, and the far right is the animals were exposed to the EGFR signal. And as you can see, there is a decrease in the overall expression of EGFR, which correlated again with another major biomarker, Ki67, where we actually saw a decrease in the number of Ki67 positive cells, indicating that we were actually decreasing the rate of cell division in these tumors.
Thank you, Javier.
Yeah.
So just a high-level summary of where we're at, where we've been, and where we plan on going. We do believe there's some significant advantages with the Halo and Voyager systems because we can get to tumors in the brain as well as other parts of the brain. Blood-brain barrier, which is a problem for many, many drugs to get to, is not an issue for our technology. So we're very confident, growing more confident every day and every time we do additional clinical research that the technology works and it can be beneficial to many, many patients, both used alone as monotherapy, but also in combination with traditional standard of care drugs. So we did meet with the FDA back in 2019. We have received what's called a humanitarian use device exemption pathway. In the medical device world, which is what we are, This is analogous to an orphan drug, an orphan indication, very rare disease. We met with the FDA in 2019. They wanted to see 2 additional data points. One was mechanism of action, and the other was a preclinical animal study in a DIPG model. And Javier shared with you both of those results of those studies. The UCSF data that you just saw that Javier shared, we are now just sending off samples of the mice tumor, the brain, as well as their other organs. We're going to do histology to demonstrate safety and again, efficacy and mechanism. We hope to have that report back sometime in mid to late June. Once we receive those results, we'll request to meet with the FDA again, probably sometime mid to late summer.
Yeah.
Assuming successful, we hope to receive the humanitarian device exemption approval for DIPG and diffuse midline glioma, which would allow us to treat patients as if we're a fully approved FDA product, which we would anticipate, if all this timing happens accordingly, sometime early 2024, we would be able to start treating patients according, assuming their physicians and parents want to use the Halo device. So that's, coming up fairly quickly. We would also commit to do a full phase 3 clinical study, which we would again be looking for enrollment centers around not only the US, but probably Europe and other parts of the world as well. On the effects of where we're at and why we want to go to DIPG and to diffuse midline glioma, as Javier mentioned, it's a horrible disease without a lot of options for parents and patients, and so it really has become close and near and dear to our heart. You saw one of our patients in one of our slides that was one of our earliest patients that we treated, and she survived over 2.5 years. So we're very encouraged that we can help, and we just believe that we are at the beginning of this journey with you. As Javier mentioned, we can test very quickly these other targets once we create the WAV files from these other siRNAs or other drugs. We can test them very quickly in preclinical models to see if there might be an effect. If you have any questions or would like to follow up with us, our emails are here. Myself, c.rivera@emulatetx.com. Javier is xfigueroa@emulatetx.com. So Melissa, with that, I will undo the slide.
And if everyone will go ahead, if you have any questions, please put them in the chat. We definitely want to take use of having them on the call with us if anyone has any questions. Okay, looks like we've got our first question from Phil Tan. Hi, Phil, great father, definitely researcher mind. He's asking, can the Halo be used to target H3K27M directly? The mutated protein or gene, this is the main driver mutation in DIPG. Should it be the main target? Is really the question.
So what's interesting is that we've tested paclitaxel against SF8628, which is an HK327M mutated cell line, and we do see direct effects on it. We have tested some of the signals against specific targets against H3K27M, and that's what we're waiting our histology results on. So we'll see if we hit it and how effective it is. Unfortunately, the clinical trials with GSK J4 were not as good as they had hoped for. They didn't seem to provide any effectiveness against it. But again, since we can actually hit siRNA targets, where it's not a drug, we can actually reduce the expression of certain enzymes. That's another target that we can potentially hit later on, and that's a great question. We really think that would be an excellent target.
And when are you expecting to have those results again?
Mid to late June.
Okay.
Yeah.
Are there any other questions? Okay, well, as they said, they've got a couple email addresses. You're welcome to email them. We're very happy that they're going to be at the DIPG symposium as well, speaking about this. And if you're in attendance, please go up and visit with them about it. I will tell you, I personally am very impressed about the no side effects, and it's pretty exciting that it can deliver the treatment constantly. And so I'm a firm believer you just keep kicking it, which that allows. So in conclusion, just thank you for everything that you're doing on behalf of the DIPG DMG community. We appreciate you joining our efforts, and we'll see you in a few weeks. Thank
you very
you.
much, Melissa.
Thank
Thank you.
you.
We'll see you in a few weeks. We look forward to it.
I think there's another question that
Yep.
just came up.
Oh. Here we go. Oh, so they want to know how this device compares to the Nativus Halo.
The same.
Great question.
So we were formerly called Nativus. We are now called Emulate Therapeutics.
Well, there you go.
I'm glad to see people were aware of that. Thank you.
Well, thank you guys.
Thank you. Have a great evening.
You too.
Take care.